Okay, so this is where we're at. Welcome back, everybody, and this is where we're at. And so in a couple of minutes, we're going to hear Sam talk about the second part of CAR T-cell therapy, and we're going to roll on. And so you have a PDF drawing assignment that's due today based on that long video that we had last week. And so you take a picture of your label drawing. You're not handing it in because you should want to keep that for study. And that's due now. And then we're going to have a quiz on the full video at the end of class today, because that quiz, that video, is as important as the videos we started our course with. The videos we started our course with focused on a specific example of a universal definition for a gene. And so we're using that gene, myogenin, over and over again, talking about the transcription factor that's primarily responsible for activating it, and talking about what myogenin does. Myogenin is a transcription factor that is, like MyoD, essential, required for muscle differentiation. Here's a little side note to that. I know we have to move on, but think about this. I've mentioned before that we have genomic equivalence in humans. All animals have it, all plants have it on our planet. It's a common, universal feature of life on Earth. And what that means is that, with really rare exceptions, every one of our cells has all the DNA we inherited from our parents. And that's a surprise. It should be a surprise because life is very efficient. And so why do cells hang on to DNA that they'll never use? And the answer is, it's very clear that there was a precedent in evolution before multicellular animals and plants evolved. There were single-celled organisms, and they already had a plan and mechanisms in place where they would have more DNA than they were using at any moment. And at the start of chapter 7, the book talks about bacterial transcription for a little bit. And that's not very important for us because it's not a course in microbiology. That's where you would talk about bacterial transcription. And one of the other reasons it's not very important for us is that microbiologists tend to use different words for the same thing that is parts of a gene. So if you've taken microbiology, you likely have heard the word operon. Maybe you heard the word cistron. And we would never use that word in here because we use words like transcription unit and cis regulatory region. Nevertheless, that's a universal explanation from our course of how all genes on our planet work, even when scientists don't use the same name. In our course, we've already run into this thing where we like to talk about the promoter and the enhancer. And they're together components of the cis regulatory region. There are some scientists, when they get going in their sort of specific method or their specific details, they'll just refer to the whole cis regulatory region as the promoter. And that's unfortunate because it's confusing when a student is learning that, oh, different people use somewhat different terms at different times. That's too bad. It's too bad because back in the 1960s, the molecular biologists that discovered and studied those bacterial genes and things like operons described the situation in bacteria way back then, just less than 10 years after Watson and Crick discovered the structure of DNA. Those bacterial geneticists and early microbiologists said, this is evidence that the bacteria has more DNA, more genes than it's using at any given time. Every cell on our planet has that. More DNA than it's using at any time. Consequently, what we learned about at the start of our course, transcriptional regulation, it's a super major theme on our planet. It's actually not surprising to hear this, that you have the insulin gene in every one of your cells. And you have it that way for every cell for your whole life. But the gene is only activated in some of the cells in the pancreas, ever. That's significant for examples like this. Elephants have lots of muscles in their nose, which is called a trunk. And there's really good evidence in the fossil record that the ancestors of elephants had fewer muscles in their nose, and then a long time ago, millions of years ago, elephants had noses with no trunks. No muscles. Yeah? And that's typical of all mammals, with rare exception. there are only a few mammals that have muscles in their noses. Rats have a few muscles in their noses, elephants do, and there's just a couple other species. Some people can wiggle their nose, but it's not because they have muscles in their nose. Those are muscles nearby that's wiggling their nose. We don't have any muscles in our nose. But here's a universal truth for all mammals. Every mammal has all the genes that we inherited from the parents in every cell in their nose. And so it's not very astonishing to see some individual mammal species that have muscles in their nose. It still takes millions of years of evolutionary change, but the opportunity is there. And that genomic equivalence provides just massive evolutionary potential. And is undoubtedly at the center of the tremendous success and diversity of multicellular plants and animals in our life. And that genomic equivalence is the fact, and then you just immediately go to, we want to look at gene regulation. Immediately, you would say, well, how are those genes regulated? Insulin's not made in every cell. Muscles are not made in the nose of every species. So what causes that to happen? And the answer is stories like we start our course with. So you see, chapter 7 is just a central part of the story of life on Earth. It's not limited to our course. It's just life on Earth. It's hugely significant. And that's true for chapter 16 as well. And so we're using that specific example from that fairly long video, that's the focus of the quiz today, because it's just central to the story of life on Earth. Here's an example of that. In animals, we start out as one fertilized egg, and then the cell divides a lot. So we have a couple trillion of cells in our bodies, right? So we're multicellular. As those cells divide, they have to start becoming different. And it's a central fact on life on our planet that for plants and animal cells to become different, say to make the brain and the pancreas, those are different cells. They don't change the DNA, they keep genomic equivalence. They change what genes are being expressed, right, like that. And there's only 2 ways those cells can become different in animals, just 2. And one of them is that the cells could divide unequally so that the 2 daughter cells in the embryo are already a little bit different. That's one. And the second one is through cell communication. That's it. There's no other possible way of making the cells different in nature. We can do that in a lab, but in nature, there are only those 2 ways. So this chapter, and what we'll have a quiz on, is a specific example of just another central story of life on Earth, not limited to students that are interested in cancer biology at all. You know, it's just gigantic. So anyway, so totally worthy for us to just hit the brakes and say, yeah, big thing to carefully have a quiz about the muscle story, quiz about the PDGF story, right? And also it's all normal. We're not looking at cancer. We don't start out looking at cancer. We're just picking a normal example that could get disturbed in cancer, and that helps us underscore our learning. Because we see how it gets disrupted, and we're like, oh my gosh, the role of RAS is really interesting in that pathway like that, right? So we're gonna have a quiz on that. Then term paper. And so let me just show you this. You should have seen that I shared, and you need to read these, but this is everybody's name, and I shared 2 Google Docs with you about the term paper. And so it's almost for sure that you'll write a term paper in this course, that the product is very similar to any other papers you've had to write. Science writing, and then a bibliography of your sources. But the mechanism we're going to do is very different, I bet, because you're going to do all your work in those Google Docs. And I'll talk about that in more detail. We'll give Sam a chance to talk, but everybody should start rolling forward on their term paper. If you are a person who likes to produce papers on the night before they're due, this system is not going to work, and it's not a good thing to encourage you to try to produce papers the night before they're due. It's good for us to encourage you to do research and start exploring and thinking about it. And the topic for the term paper is set out in the syllabus. And then I made a video and posted already on Brightspace how to write the term paper. The topic is just you're gonna pick one of these receptors, any one. You can pick the estrogen receptor, you can pick the PDGF receptor. or any other ones. Well, the CAR T cell receptor, you could talk about that, right? Any receptor, and you write a paper about it based on the description and syllabus. And that's just, again, focusing again on how important the story of cell communication is, not just for cells to become different, but right now, all the thinking we're doing is cell communication. Our nerve cells, our neurons in our brains are communicating by neurotransmitters, which are signaling molecules. We'll learn about one of them a little bit. We'll learn about dopamine in this chapter and its role as a neurotransmitter, but also its role in broader cell signaling like that. So that's that. And then I think with that, back to this one, because then our exam is a week from Thursday. And you know what it's like, and I sent out a exam from a previous year. And I described in the bright space how I'm not giving an answer key, and this is the only old exam I'll share, and me doing it for a specific reason, so you can see what's coming. After the first exam, you know what's coming for the second exam. It'll be the same format, and you'll know what's coming for the final. But you really don't know what's coming in this course for the first exam, even though we've had quizzes, right? So really need to do that. Now, you're not expected to be able to answer all those questions. I don't provide an answer key, because I described that I just think that when students get the answer key, they just kind of scroll through the exam. It'll be good for you to work on it, because one of the things you need to practice doing is recalling knowledge and practice being ready to answer questions on the exam. You know, if a friend of yours says, oh, do you like that song? And you're like, I know that song, I like it really, I like it a lot. And if they say, well, go ahead and sing it, then you're like, okay, all right, you know, like that, right? You're really being tested on whether you know the song or not. And that's what our exam is like. And so there's a feeling like when you hear a song, oh, I like that song, I've listened to it a ton of times. But did you actually learn it, right? And so that's deceptive in learning, right? I've said it, it's pretty obvious. You have heard the instructions of how MyoD activates myogenic. But can you say it? That's really the key in a test. So the old exam is intentionally left with no answer, so you can work it out. And we could meet and talk about questions and things you have, and you can bring them to class and stuff like that. They'll be fine. Yep, okay. So that's that. And let's jump in, give Sam a chance to give his presentation. And remember, the situation is we're just changing the instructor on a specific topic. That helps you learn about it. Because you can think, oh, that's that thing that Sam talked about. And I don't really get why Sam did that. He's in the class. Like, is he going to do well on the test? Because there'll be questions on the test about CAR T cell therapy, you know? And we're just not going to worry about that. Yeah? You know? Don't worry about it, because you're not competing with each other. You know, that's not how the grades in this course work. So this is Sam. You're going to talk about another piece of it that should be helpful at this point.
Thanks, Dave. Good morning, everybody. Happy Tuesday. So I'm Sam again. I sit in the seat Dave's sitting in usually. Another fun fact to know about me, I lived in Philadelphia for 7 years before moving to Maine, and have a very, very deep love for that city. So if you guys have ever been there or like want to talk about it, I'd like honestly love to talk about it. It's like a weird thing of mine.
Is that where you're applying medical school?
I will be applying there, but I'll be applying all over the East Coast. So, yeah. Okay, so second talk. Today we're really going to be talking about the idea that the CAR is a receptor, and receptors set off cell signaling pathways. And really we're going to focus, after our lecture on Thursday, I'm really focusing in on integration, that idea that cell signal pathways integrate with each other and themselves to produce complex results. So that's where we're going today. Okay, so cell signal pathways. And I was here to talk about transgenes. The CAR is a transgene, and I'll come back to talk about cellular responses as well, specifically to the CAR. So quick recap, right? The CAR, the chimeric antigen receptor, is a chimeric receptor. So these are all things, made of different protein domains that exist naturally in the human body, and we're combining them together to create something that has never existed before in nature. Yeah, Dave?
Are we combining the domains of the protein together?
No, Dave, we're combining the genes. Thanks for the clarification.
Thank you.
We're combining the transcription units of the genes with a promoter that is normally expressed in these cells. Yes, Dave?
what specific region of the transcription unit are we using in this work?
Depends on the gene. So some genes we're using if I understand your question.
You don't. We're using the coding region,
Yes.
right? That's the specific part that codes for the protein, and we want to use those portions and stitch those together.
Right.
Yep.
And we're using subsets of the coding region.
Yep.
Yeah.
Portions,
That
yep.
cancer, right?
Like that
That
cancer.
was the whole point, yeah. Okay.
Yeah, like that cancer, exactly. Okay. So this is a photo very similar to the one Dave has been showing us. We're going to come back to this a few times, right? The idea, a ligand, a signal, they call it, we can call that a ligand a lot of times in biology, binds to a receptor, and then that phosphorylates something. Usually it's phosphorylation. Sometimes phosphorylation turns things off. Sometimes it turns things on. But we can usually think of phosphorylation generally means turn on. And it turns on, which turns on, which turns on, and then the cell does something. So this is a photo from our textbook. And we're going to, again, come back to this idea, right? So this is the idea that you can put multiple of these signal pathways together, and they start to talk to each other. And so, we see here, and this is, here's our RAS, RAF, MEC, ERK pathway that we just talked about we will have a quiz on. And here's something called the mTORC pathway. The names of these aren't super important, and what they do isn't super important. But what is important is the idea that these two pathways are talking to each other, and that they have relationships here. So this ERK protein inhibits this protein, but activates this protein. And so what that means is that as different signal cascades activate, they can produce complex results. Integration. So so this is a normal process for T cell activation. T cells, part of the immune system. We're specifically interested in the T cells called killer T cells, and they kill. They kill other cells. And so normally they're like this lighter. You have to push in the button and squeeze the trigger to produce a flame. And so this is an image, this idea that this T cell here in green at the bottom, both need signals from this immune cell and signals from this immune cell that binds to different receptors and activate different pathways to integrate to achieve activation.
Can you define integrate one more time? It's just putting two things together, kind of like the chimera thing.
No. Integration is a property of the signal transduction pathway.
Okay.
And it's the idea that when I'm watching a movie, I'm both taking in visual information and auditory information and combining them together to understand the movie. And so for the signal transduction pathway for T cells, deep breath, what we want to take away from this slide is that you have multiple receptors binding to multiple ligands that then interact, all these different arrows, to produce a coherent response. And some of these pathways interact on the same proteins, right? And so right here, we have this protein, this transcription factor, being activated by both this protein and these proteins. What their names are doesn't matter. And that's the integration, right? That we have these multiple signals coming into the cell that produce a coherent response. And so the one piece here's our RAS and then ERK1/2, right? Here's our RAS, RAF, MEC, ERK pathway again, coming in from this one right here. An important question. Thank you. So we looked at this slide last time I was here. These are the different domains we're going to put together, right? We're taking the transcription units for different domains, putting them together in a transgene, and expressing that transgene in a cell. And so what that means, and what I like about this piece here, is over here on this side, right? We have this outer bit coming from here, and then these are the inner bits coming from over there on the far left. And those produce the one coherent signal, right? We have one binding, multiple signals being activated at the same time. And so what we're doing is we're taking the safety off that layer. We're just ready to light things on fire right now because this patient has cancer. And we just need a T cell that's going to kill it.
So it's Doug. Like the example of like locking the gun up so Doug doesn't find it.
Yeah, it's Doug. Doug's just playing with guns.
Let's just do this old time. Can you just pause, or are you
Yeah.
pretty nervous to get through this?
No, I can pause. We can
All
take
right,
a ton of
let's
time.
just set this up, okay? So there used to be a professor here named Tom Knight, biology professor. He's very popular, and I would ask students, why is Tom so popular? And Tom would do this. He would be like, okay, so now we're going to set up a scenario, and we're going to hear this from Sam Moore. We're going to have students portray this scenario. So Joseph is going to come up here, Anna's going to come up here, and Sam's going to be up here. All right?
Wait.
Yep, come on up here. And we're going to do this, where Anna is a burglar and is playing a burglar and cancer. Joseph is playing a T cell and is playing the homeowner. Sam is playing the detective. So Joseph is sleeping. I should try to stand out of the way. Joseph is sleeping. Anna comes in and steals stuff and leaves. All right?
Stay.
So, okay? All right? So that happened. All right? So now the detective comes to Joseph's place. I think I failed to mention, the detective is the doctor. And the detective says, Joseph, you have cancer in your body. You can go ahead and say that.
Joseph, you have cancer in your body.
You had that. You had cancer in your body, you had a burglar, and Joseph's like, I know, I'm in trouble. I need to defend myself. I need to protect myself, right?
That's right.
That wasn't good, yep.
Yeah, okay?
Yeah.
So now you say, do you have a gun in your house?
Do you have a gun in your house?
I don't, no.
Wow.
No, you say yes, it's killer T cell.
Yes, I actually do, yes.
Yeah.
Okay.
And you say, I'm a doctor, I'm a detective, I can help you protect yourself. Give me the gun.
I'm a doctor detective. I can help you protect yourself. Can I have your gun?
You give the gun.
Sure.
So you, the doctor, takes the T cell guns out of the patient's body and comes over here to the lab
That's right.
and takes the gun and fills it with bullets and turns off the safety, because that's what the car is. We've removed all the safety and protection. And then he goes back and gives it to Joseph. And the last part we're not going to depict because it's brutal, right? But now Joseph is sleeping. Detective leaves. Joseph is sleeping with a loaded gun, ready to go. And when the burglar comes in, the cancer cell is in Joseph's body, and Joseph shoots the burglar. We're not going to depict that. It's okay for us to depict the next thing. And the next thing is, I'm another healthy cell in Joseph's body. And here's the danger. Joseph shoots me by accident because he's got a loaded gun. And I'm like, Joseph, it's time for your PDGF quiz, and boom. Yep. So that's the situation. It's like that, the cancer. So you can sit down again. But that's how this other teacher taught, and I've never done that, and when I'm sitting down, and listening, it's just dangerous, because I'm like, oh, I have an idea. Let's make a play. So again, you sit,
I have a question though. So you said that he's then going to attack you. How do we turn off this chimeric protein to not attack healthy cells afterwards.
It's the dangerous game they're playing, and Sam's trying to talk about that.
Okay.
The dangerous game is the reason Joseph had the gun, the killer T cells, with the multiple signaling pathways, is to make it safer for the people who aren't the burglars. The other family members, the neighbors, those are just the other cells. And if Joseph shoots them, that's called an autoimmune disease. And that body has to be really careful. It has to be ready to fight off an intruder, a foreign thing, but also to be careful not to hurt itself. And so that's the dangerous thing the doctor's doing here, is they're saying, let's take the killer T cells off, and we will put all these receptors together into one. So we can just have the gun is loaded and ready to go. And it just takes one thing, like one strange noise, and Joseph is just like, boom. And it's dangerous because they could attack the body. So that's the danger there, and the anxiety when the doctor puts them back in is, well, that's gonna go after that burglar, but it's also dangerous for the rest of the cells in the body. Yeah, Ken.
So are we basically just creating a more efficient way to attack an antigen?
Yeah, exactly, but also sort of, hopped up, ready to go, dangerous situation. It's dangerous, right? The danger is, you don't shoot any intruder, but you can shoot, even some of your family members by accident. That's an autoimmune disease, right? And I know that's a brutal example, but, right, burglar, cancer, homeowner, body with a T-cell killer gun that has an unsafety, no bullets, locked up, detective, doctor. There's been a problem. You've had a burglar, cancer. I'm coming in, I'm gonna take that gun out of here, I'm gonna modify it, that's the car, make it just like really ready to go, put it back in your body. And you're just gonna shoot the burglar, but there's danger in that. So I don't know, I wouldn't say maybe more efficient, but more trigger happy. That's what the T cell becomes. It's just ready to rock.
Yeah.
And it's dangerous. Yeah.
And that's why we I think to kind of step in here and like use this image again to talk about exactly what Dave is talking about, right? T cells kill. And if they just kill anything they come into contact with, your body would kill itself. And when it attacks certain cells in your body, that's an autoimmune disease, right? And so normally it takes two signals, at least two signals, to activate a T cell.
And integrate.
To take two signals that are then integrated to activate a T cell. Right? Here we have three. And then we're going to combine all three of these signals into one signal molecule by combining the domains, the intracellular domains, through the chimeric antigen that is the transgene. Right, we're going to create this novel protein by combining the DNA to create this new receptor that doesn't have any safeties. Right? There's no safeties here. It's just go, just kill. So it binds, right? And it binds. It's got a little extracellular region. It binds to the cell, the cancer cells, and that sets off the signal transduction cascade.
So you make a CAR T cell that has the T, like that has a CAR receptor, so there's like double CAR? I was confused by it because it says CAR T cell and then it says like the CAR.
Yeah, so this is the CAR T cell. That's what we call the cell.
Okay.
Yeah, that we've taken out of the body, modified in the lab, and put back into the body. And the CAR, the chimeric antigen receptor, that's the specific protein that we have engineered this T cell to produce. Okay, types of receptors. This is really getting into some weeds on chapter 16. G protein coupled receptors. So far we haven't talked a lot about those in this course. They're in the textbook. I'm not going to talk about them today. Receptor tyrosine kinases. That is our quiz and homework for today. And then what are called non-receptor tyrosine kinases, which are, which are similar to receptor tyrosine kinases. Normally, cell membrane, a receptor tyrosine kinase has this inherent catalytic activity. right, where it's the two dimers, the two strands coming together, dimerizing, that then phosphorylate each other. In the non-receptor tyrosine kinase, you still have that phosphorylation action. It's just performed by a different protein, signaled by this little meatball. Right, so we still have phosphorylation of the receptor. It still attracts molecules that can bind to it that then sets off a cascade. So it's a variation on what we drew and studied for today. Okay, deep breath, everyone. There's a lot of weeds on this next slide. Don't worry about the weeds. What's important here is that these different domains come from different receptors, as they already exist, and what I like about this one here on the left is that you can see the phosphorylation, right? That we have these domains that we're combining together. Coming back here, a few names. I'm gonna name these things for you right now. The names are not important, but I need to tell you the names so that you can understand the other graphics, because that's how scientists refer to them by these names. So the things we're going to talk about today are CD3 zeta, horrible name, I'm sorry, 41BB, and CD28. Okay. Names aren't important. These are just different things.
Well, those are the three natural receptors where the doctors get three of the domains from.
That's right. Thank you, Dave.
So that's what you're talking about.
And that's what I'm talking about.
The four genes that get combined to make the car from the first time you visited, that's the actual names, the three of them. And those three names will not be on our exam. But we are learning the types of receptors, the classes. So one is a receptor tyrosine kinase, one is a non-receptor tyrosine kinase, and one is a G protein coupled receptor, which we'll have examples from our book on the exam with specific examples from those categories.
Thank you, Dave. The names aren't important. The reason why I'm telling you the names right now is so that I can then point at this and say that, do you remember that domain we combined together to create the car? It is part of this receptor family. So I'm going to write those up here so that you can keep track of them as I'm going. I know we're in the weeds. I don't think we're going to stay there. It's okay. 41BB is part of the tumor necrosis factor superfamily. These are kind of cool. There's a section about them in our textbook. They activate, the piece we want to pull out of this is it activates the mTORC. That's another pathway. Activates the mTORC pathway. Right? We don't need to know what that does right now, except to say that it is part of a pathway that is going to be combined with other pathways as part of the integration that we're talking about today. Oops. Oh, here we go. Why am I? Here. These are the pathways we're integrating, right? Here it is, 41BB. Pretend that says CD3 zeta, CD28, right? These are the different domains that are naturally expressed on, these are proteins that are naturally expressed on killer T cells that we're combining together in the car to create the novel receptor. Okay, more deep breaths. This one is really weedy. I'm going to point out the things we need to know about. And the main idea here is that here we're really going to get to see the integration of various signals. Okay. So here's one of the reasons why I've been talking about those names, just so that you can recognize the different intracellular domains of the CAR. Right? Here's that CD28. Here's 41BB. Here's CD3 zeta. And here we go. We have the different pathways that are being activated. over here, RAS, RAF, MEC, ERK, right? We know that pathway. Second messengers, we talked about that on Thursday. Here's PI3 kinase going down here. And I think that's all of our known friends that we have right on this slide. Okay, and then this is the continuation of that same slide we were just on, right? Here's the MEC-ERK of the RAS pathway we were just talking about. Here we have this whole mess, PIP2. We remember that from second messenger conversation. To DAG, to IP3, right? Mobilizing intracellular calcium, which then causes a further cascade, right? So that's our second messenger cascade right there. And then the piece that I'm going to pull out to kind of talk about this idea of really of integration is that this cluster right here with the yellow, the blue, and the green is also right here, right, on different sides of the same pathway. So we have multiple signals coming from these different domains that are being combined to produce a coherent cellular response. And I told you we would come back to this. Here we are. This is the cartoon, simple version of what we just were looking at, the details, the weeds. And I think this is useful because, right, this idea that these pathways are talking to each other. We're creating a receptor that activates multiple pathways at the same time. Those multiple pathways are part of different receptors normally as a safety mechanism. You have to push in the button on top of the lighter and squeeze it to, when I was 5, I was taken to the emergency room because I was playing with matches. And apparently they separated my parents and were interrogating them about whether or not this was child abuse at home. So the lighter story felt really appropriate, right? Because we don't want kids to burn themselves with lighters or to accidentally set your house on fire. But our patient has cancer. So we're making a lighter that has no safety. It just burns. You know, this is California and we're PG&E. The way we create that is by making a novel protein, by combining together the transcription units, parts of the transcription units of all those different genes that we want to combine together and attaching them to the regulatory region of a protein that is normally expressed in T cells.
Great. Thank you. All right. So we'll take this question for Sam, or maybe or maybe I'll jump in and answer questions. So
okay,
Sam can take a break. But first, let me say I apologize about using a gun analogy. Guns are dangerous and not good to be talked about like that. It just fits really well, and the car is making this dangerous situation like we portrayed in class. I think it's just a memorable example. And that's, again, how this other biology professor apparently used to teach. I never saw him teach, but students got a kick out of that because it's kind of unforgettable. Bad analogy because of the danger involved. But accurate, I think, right? Okay, but questions about that. Let me just show you this. So it's nice to see that and then recognize, no, you do not have to learn anything like that. There are no pages like that in our book. The pages in the book have stripped down versions, so we could point to specific pathways like that pathway that uses the phospholipase C to activate and get the triple messenger cascade going like that. And so we know about that. And in that example, we're going to learn, I think, a total of 5 signal transduction pathways in our Chapter 16 learning. Because more than that, we're going to amplify 5. We've already learned 3. One is the RAS MAP kinase pathway that's used by a lot by receptor-tyrosine kinases, and we'll learn more about it as we go along because it has a lot of oncogenes in that pathway. RAS, MYC, we'll learn about another one, after the first exam. Then the second one is the PI3 kinase pathway, and that phosphorylates that PIP phospholipid in the membrane. Becomes a way of activating more proteins, especially a kinase called AKT, because AKT phosphorylates FOXO. And so we learned a little bit about FOXO. FOXO is a transcription factor that is turned off when it's phosphorylated. It gets prevented from going into the nucleus and doing its job. And then the third one from the pathways of the receptor tyrosine kinase that we learned about is the one that's not in our textbook, but I showed the figure, and you just have to learn it just from the class meeting that we had. And that's that PI3, that phospholipase C. That's going to cut. So again, our second pathway and our third pathway are both focused on PIP, that phospholipid in the membrane. The first one is phosphokinase C. It's going to phosphorylate it. The second one is phospholipase, or PIP3 kinase, sorry, and then phospholipase C. And so one adds a phosphate to it, the other cuts it in half to produce IP3 and DAG. Like that? So those are three. Then when we get to talking about the G protein coupled receptors, we'll learn two more pathways. Yep? Yeah.
I was going to ask you about the other two pathways. Are those also involved in cell healing? Like, is that the goal, is to like to stimulate like the cell cycle?
So the answer is that these other pathways sometimes are used to integrate and add together to make the proliferation, to stimulate proliferation, because that's also a dangerous game to say, oh, you go ahead and divide. So that usually uses a couple of pathways. But then also, the same receptor can be used in different contexts to stimulate very different things, and the same signal transduction pathway can be used in other contexts to stimulate different things. So we want to be careful not to attach too many examples to a specific pathway and what it does. We'll attach a couple, and one we'll attach is that when we think of the RAS MAP kinase pathway in our course, we'll think just about cell cycle stimulation. It's true, it can do other things in other cells. It does, like that. Yep. Yeah. And the next meeting that Sam's going to come to is to talk about, well, what does the T cell do when it gets activated? And that's called the cellular response. And in our course, we're mostly going to emphasize new gene expression. It's going to turn on new genes. But there are also things that happen, we'll learn, that aren't new gene expression. So cellular response can include new gene expression or other kinds of cellular responses, or both. Yep. Other questions you have about this? Okay, so hopefully you can see it's a package, right, where we see this CAR T cell therapy pops up because it's got something to do with transgenes and building that CAR transgene and our knowledge of the organization of DNA and genes. Then it pops up because it's a receptor, and it is a combination of receptors that normally activate different signal transduction pathways, and those get integrated because it's a dangerous thing that's going to happen. Yep. And then the third visit will be cellular responses. Our favorite cellular response in our course is going to be stimulating the cell cycle, so stimulating cell growth, because that gets disrupted in cancer. Cancer is out of control growth, out of control cell cycle growth. So in the video, I talk about the skin cancer patient. Her mutation is in this pathway. PDGF receptor is a receptor tyrosine kinase, and it activates RAS, MAP, MYC in under a scab. Every scab you ever have, that's what is one of the important things that's happening. Back to your specific question, I don't even know. There's likely additional pathways that get activated.
We always say that it's like we phosphorylate FOXO so it can't do its job, but like what is FOXO's job? Like you make FOXO.
Yeah, FOXO's job is crazy. FOXO's job is out of control crazy. There are orthologs of FOXO in every animal. And FOXO plays an important role in the rate of aging. So I talked about that briefly on the first day of class, and I use it as an example because molecular biologists really don't care much about what is done. They really like the nuts and bolts details of just the mechanism of the pathway, the mechanism to activate it. So a molecular biologist would be like, oh, whatever, so Joseph is going to use the CAR T cells to fight cancer. You know, that's great. I'm just hung up on the details of the CAR and how it's activating the T cell, right? Like that. So yeah, but we use FOXO as an example. When FOXO gets to do its job, the rate of aging slows down. FOXO was originally discovered in a really simple model organism called a nematode worm. And in the nematode worm's life cycle, it has two options. If the environment is beneficial, so plenty of food, good temperature for growing, and not very crowded with other nematode worms, the nematode grows up fast and dies after about 2 weeks. In that animal, the FOXO is inactivated and not allowed to do its job. However, if the little nematode worm is born and is growing in an environment that's not favorable, not beneficial, so not very good food, not a very good temperature, and crowded, it actually goes into kind of a hibernation state. And it stays in that state for months. And it has little chemical sensors detecting food, temperature, and then the worms all make a chemical that's used like a pheromone to detect the presence of the other worms. So if it's a high concentration, it must be crowded, not a good time for me to come out of hibernation, I'll wait. And the animal can wait for up to 5 months and then come out at any time along the way. And the surprise of that was the scientists discovered 30, 40 years ago that to do that, the animal has to actually slow down its rate of aging. And it does that primarily in that real simple worm by letting FOXO into the nucleus. And FOXO is a transcription factor, and you immediately say, wait, what are the targets that it activates? And there are a couple hundred, and all of them slow down aging. Just remarkably, like it would give you the shivers to see that happen. We don't have that in the human life cycle. But we have FOXO, and we have different alleles of FOXO in humans. And there's a correlation between the allele of FOXO and just especially certain families that have an inherited tendency to live really, really long, over 100. And there's a correlation with the type of FOXO allele they have and how long that family lives. That's not the central single basis for aging in humans. Raise your hand if you're taking, there's an aging seminar that Doug Curry's teaching right now. Raise your hand if you're taking that. Yeah? So a few people are in there. So we're not going to talk about aging here, partly because whatever, you know, as cool as all that sounds, whatever, what's cool about FOXO is that it's on that PI3 kinase pathway. And when the PIP gets phosphorylated, it binds to a protein that activates AKT. And AKT phosphorylates FOXO. So that's what's cool for us. Yeah, it's kind of remarkable. But we can ask questions about that because you're just like, whoa, I want to learn more about FOXO. I want to sign up for Doug's seminar. Yeah, Al.
I had a question about something from the video.
Yeah, absolutely. Oh, yeah, because we're trying to get ready for our quiz. We're going to have our quiz here in just a few minutes. Yep.
So I was just like a little confused on some of like the details of the DFSP. So there is the chromosomal translocation between PDGF and the cis-regulatory region for skin collagen.
Yeah.
And it's between 17 and 22. Does that mean like it gets cut at 17 on the PDGF and at 22 on the collagen?
What it means is that the first thing is that because of genomic equivalence, the skin cancer patient had all the genes. And one of the genes they had, all the genes in their skin cells, their whole life, all the time. And one of the genes they had is expressed always in the skin cells, because it makes a protein called collagen that makes our skin tough. Collagen is a connective tissue protein, and the collagen is secreted. Collagen is always secreted from cells. And then it gets kind of twisted together like strands of thread to really hold our tissues together. So that gene was there for sure because of genomic influence, and it is normally and always made, and it gets secreted. So that's always there. Then the other gene we're interested in, because she has a problem with it, is every one of her cells, every one of our cells in all our skin cells has the PDGF gene, but it is not active in our skin. It is active in our bone marrow in the megakaryocyte cell. That megakaryocyte makes platelets, and it puts the PDGF protein in platelets, and platelets are like a little suitcase that are constantly floating through our body. They're just little blipped off parts of cells. They're like a little protective package that can release their molecules when needed. And the most famous thing that platelets do is they have clotting factor. And when there's a wound, that's who releases the clotting factor from this platelet suitcase, and it clots the blood. The same platelets release PDGF, platelet-derived growth factor. A growth factor, because where there's clotting, there's a wound. We should just, at the same time, start stimulating healing, like that. So she has that in her skin cells, but is not using it. Unfortunately, she starts using it because of a mutation. And the type of mutation is called a chromosomal translocation. And a chromosomal translocation is like this, that there can be situations where the mutation, where a chromosome gets torn in half. Both of the strands of the DNA break. And that happens rarely, but it's called a chromosomal break. And the body has repair mechanisms, DNA repair mechanisms, to just glue that back together. So let's say you drop your cell in Molec book and it just tears in half. A chromosome, a book torn in half, glue it together. Now let's say that you drop your backpack and 2 of your books get torn in half. Chromosome 17 book and chromosome 22 book. And you glue them back together, but because there were 2 broken at the same time, you make a mistake. That's what happened in her cells that caused her cancer. By chance, she broke 2 books at once, 2 chromosomes at once, 17, 22, and she glued them back together, and she glued them back wrong. She glued the part of her cell Molec book together with her math book. And right at the glued in point is where the collagen gene were and the PDGF gene were. The regulatory region and the transcription unit of the skin collagen gene happen to be on 2 pages. The regulatory region was on 1 page, and they had to put the transcription unit on the other page. And that's right where, unfortunately, her book tore. And then her other textbook for math had the same situation, but it was the PDGF gene. The regulatory region was on 1 page, had to put the transcription unit on the other page, just ran out of room in the textbook. So it was like that book got torn there. And when she stitched them together, she just actually glued her cell Molec book with her math book. 2 chromosomes got glued together incorrectly. That's called a chromosomal translocation. It's a mutation that's very rare, but it happens. That's the kind of mistake it is. And when she did that, she built what we would have called, so up till this point in our course, a transgene, right? She took, like when you make a glowing muscle cell, she took one cis-regulatory region and glued it to another transcription unit, and now she has this hybrid gene. We don't call it a transgene because it wasn't built in a lab. It's just a chromosomal translocation, and when you zoom in on what pages are affected, you're like, oh my gosh, what an unfortunate place to tear these 2 books apart, because we have the collagen regulatory region glued onto the PDGF transcription unit, and she now makes it in the skin cells. She should not make that protein in the skin cells. Yep, and then, but you're still, and Just has a question too, but you're still clarifying?
One last thing, so then from there, it makes the chimeric protein, like that gene starts creating the chimeric
Yep,
protein.
so that mutation is just brutal on the unfortunate. Like that, like where you want to just go for a walk when you hear exactly what happened, because these are random breakpoints. And the situation is this. Normally, the PDGF gene is made in the megakaryocytes, and it is not released. It stays inside the cell. And if you remember back from BIO409, intro bio, that's because it does not have a signal peptide on it. A signal peptide is needed for a protein to be targeted to complete its synthesis in the rough ER, then go through the Golgi and vesicles and be secreted. PDGF is not normally secreted. I mentioned already, collagen is. So when you get to the coding region of the collagen gene, the first amino acids are about 20 amino acids that are called a signal sequence or a signal peptide. And they target the newly synthesized collagen protein for secretion. That's introvio stuff. Her breakpoint is not just between the regulatory region and the transcription unit like I've described so far. It's like that, so we get it expressed the wrong way. But the breakpoint is actually within the coding regions. So she's making a chimeric protein that has just a little portion of the collagen protein connected to the PDGF protein. Because what that brings is that protein will get secreted. It will get made in her skin cells and it'll get secreted. And it needs to because the PDGF receptor is on the surface. So we need to have that protein actually released, not just made in the cell, but released. And so the actual specific breakpoint varies slightly, and that's shown in the video with some purple and red, showing that in individual patients there's a specific breakpoint, but it's always in the coding region, actually, of both the collagen and the PDGF. And it's in the coding region, and the coding region is made up of triple letter codons, and the breakpoint has to be at very specific locations so that we keep the same, what's called reading frame, or codon use from the collagen into the PDGF. So it's just a horribly unfortunate mutation. Just like, oh, wow. Like that. I mean, you know, take all of your learning that you've done and think about how tough those breakpoints are. Those happen in our body. Most of them are just random where you join the chromosome back together and you're fine. Or you even have a chromosomal translocation and it's in between genes, or it's within genes, or it's even within those two genes. But the matching is imperfect, so you don't make the correct protein. And she makes this hybrid protein that's just brutal. Yeah, oof, right? So we have those. We have already accumulated tons of mutations in our bodies, you know, that just aren't as problematic and unfortunate. It's just like that. And when you get down to it, those cancer stories are always like that. We're just, darn it, you know, talk about unlucky. The other piece of that is it's just one mutation, you know, like it's not like, you know, she was laying out, you know, tanning on the beach, smoking cigarettes and eating, you know, junk food, you know. It's just like a very, very unlucky mutation of all the mutations, you know. Woo! Yeah. Just had a question too. And then we have to take our quiz.
I guess my question was, is there specific amount of versions of those genes that make them more likely to break and stick in places they'll stick together to create that hybrid protein? Or?
Yeah, at first round people would say those breakpoints occur pretty randomly. But it turns out they're not perfectly randomly. There are some like more likely areas to break like that. And I don't know the specifics. Is that a kind of a more likely place to break than others? You know, so I don't know that answer, specific answer. But it is true that they vary. Okay, so now you want to turn off your cell phones, and we're going to do our quiz. And it's on both sides, front and back of this one piece of paper. So make sure you check it out. And then when you're done, you can head out. I'm going to post a thing. I'm going to post a Brightspace thing about an event that's coming up in April that we would have to register for this week. And it's about students who are interested in healthcare professions. And so take a look at this and think about it if you're interested in the healthcare pathway. Because it's not for everybody. It's in for a certain set of students. But it's a crazy, even if you have, it's on a Thursday afternoon, and even if you have a course that afternoon, if you fit what they are hoping to get over at Maine Health for this event, you should skip the class. And you should tell your instructor, hey buddy, I'm skipping. And if you want to talk to anybody about it, talk to Dave Champlin. Or you can do that ahead of time and say, but I get permission to skip, and if you want to have details of why I'm skipping, you can talk to that Dave Champlin. Okay, so both sides of the paper, and then when you're done, you can head off.